Hello students, now we will start with new topic, that is proof of DNA as genetic material. Two different types of experiments are done by two different scientists, that is Griffith and also Hershey and Chase. Griffith has taken two different strains of bacteria and has done one experiment and proved that, and he tried to prove that DNA is genetic material, but he couldn't prove, he couldn't explain his experiment completely. Later, other scientists like Avery, MacLeod and McCarthy has concluded and finished that experiment and has explained this proof or the experiment done by Griffith. Other type of experiment is by Hershey and Chase. Those people have taken bacteriophage, that is T2 phage. Bacteriophage means the virus that infects the bacteria, and they have taken this bacteriophage and also one bacteria, and they have also included radioactive elements like sulfur and phosphorus and has done their experiments. We'll first see about the experiment done by Griffith. We also can write this one as Griffith experiment to prove DNA as genetic material, or Griffith experiment of transformation. He started his experiment in 1928 by taking two different strains of bacteria. Griffith selected Streptococcus pneumoniae, for his experiment, and out of that he has selected two different strains. Two strains. What is first one? First one is smooth strain, means one type of Streptococcus pneumonia bacteria. This smooth strain has capsule. What is meant by capsule? On the cell wall, means on the peptidoglycan, there is some extra covering called as glycocalyx. If that glycocalyx is hard, then we call it as capsule. If glycocalyx is loose or slimy, then we call it as slimy layer. Capsule means glycocalyx is hard. Now, it exists in smooth strain 1 form, smooth strain 2 form, smooth strain 3 types of forms. Because of presence of capsule, Streptococcus pneumoniae smooth strain is considered as virulent or pathogen. Is virulent means it causes infections, or pathogenic. Next, coming to other type of strain, that is rough strain. Rough strain has no capsule. Now, it exists in again 3 different forms, that is rough strain 1 type, rough strain 2 and rough strain 3 types. These are non-virulent or non-pathogenic. Out of these 2 different type of strains, Griffith has selected this S3 type of pathogenic one and R2 type of rough strains. Griffith selected S3, that is smooth strain pathogenic, and rough strain 2 type of strain for his experiments. So he initially injected a rough strain into a mice. Mice did not die, it survived. Again later he injected smooth strain, that is S3 type, which he selected for his experiment, into other mice. Then mice was dead. When he mixed those both strains and then injected, again the mice was dead. So we'll write that one point by point. When rough strain 2 was injected, live rough strain 2, nothing heat killed or anything, injected into mice. Mice survived. Or mice is alive. Second one, when smooth strain, a third type, which is alive, this also alive in condition, was injected into mice. Mice was dead. Next, in the third step, heat killed smooth strain 3 type was injected into mice. Again, mice is alive. In the fourth step, heat killed smooth strain and alive rough strain 2 together injected into mice. Now, mice was dead. But Griffith has done these experiments, but the biochemical nature of transforming substances was not explained by Griffith. It means that Griffith is unable to tell which biomolecule is responsible for death of the mice. Griffith is unable to explain which biomolecule is responsible for death of mice, and also which biomolecule got transformed or transferred into the toxic type of biomolecule. We can also write this one as biochemical nature of transforming substance was not defined or explained by Griffith. Later, in year 1944, it means after 16 years of Griffith experiment, 3 scientists involved in the continuity of this Griffith experiment and has given final conclusion. Means after 16 years, 3 scientists continued Griffith experiment. Who are those 3 number of scientists? One is Avery, second scientist is MacLeod, third one is McCarthy. So they have taken smooth strain 3 and rough strain 2, but here smooth strain 3 type is heat killed. So they have directly taken the mixed type of strains. By one, pathogenic one is heat killed, but rough strain is alive. Avery, MacLeod, McCarthy initially observed death of mice when they injected heat killed smooth strain 3 plus Li rough strain 2. They also experimented with different types of enzymes using with different enzymes. So what are those enzymes? So first case, when proteases are added, to smooth strain 3, that is heat killed I am writing like this, HK, plus rough strain 2, that is Li, then mice got dead. In the second case, when they added lipases, which break down the lipids, are added to smooth strain 3, that is heat killed, plus rough strain 2, that is Li. Again, mice was dead. Next, in the third case, they have added DNases, are added to smooth strain 3, that is heat killed, plus rough strain 2, that is Li. Now, mice survived or alive. From this three situations, they have concluded that DNases has broken down some of the transforming toxic material that is harmful for the mice, so that mice was survived. It means that DNase, which breaks down the DNA, has broken harmful or DNA in two pieces, so that DNA of rough strain 2 did not get transformed into pathogen. Did not get transformed and was destroyed because mice survived, and this is the clarity that DNA is the genetic material and is responsible for transformation. So, what is conclusion under Griffith experiment? Conclusion is DNA is considered to be transforming substance from non-pathogen to pathogen. So, conclusion is DNA is the transforming substance. And responsible for death of the mice. Now we will see about Hershey and Chase experiment. So these people have taken T2 phages and E. coli for their experiment. E. coli is an anaerobe gram-negative bacteria. Let's see the normal structure of T2 phage. T2 phages, their complete body is made up of two different molecules, that is proteins and DNA. This is called as head part, this is called as neck part, this is called as tail, and this is called as collar. Head part has proteins or protein coat, and this is nothing but DNA, which is genetic material. Now, they initiated their experiments. They first cultured E. coli in two separate mediums. They next added T2 phages and grown E. coli, because E. coli, first of all, they need more quantity in number. Next added T and grown E. coli along with T2 phages. But what happens if we grow both of them in the same medium? Will they grow? No. T2 phages will infect the E. coli and they will increase their number, but the E. coli will be dead. They will carry on a process called as lytic type of cycle, means breaking of the host E. coli and increasing the T2 phages number. Now we are going to prove that the genetic material inside T2 is responsible for death of this E. coli. During this growth, T2 phages infect E. coli by different steps. That is, adsorption means sticking on the outermost part of the host cell. Adsorption. In the second step is injection entering into the E. coli body. In the third step, replication of the T2 phages, not the E. coli. Means now here T2 phages are increasing their number. In the fourth stage, lysing, breaking the host cell. That is called as lysis. In the two different mediums, when they are taking here, in the next stage, the food material that is used or the medium is included with radioactive sulfur and radioactive phosphorus. You can write in two different mediums. One with radioactive sulfur and second with radioactive phosphorus. Radioactive sulfur, here we represent this one with S35, phosphorus is P32. Now we will discuss this one in detail. Let's take one medium or first medium. We are telling this is included with radioactive sulfur, which is indicated with S35, means this element releases the radiations. Second medium, it includes radioactive phosphorus, which is indicated with P32. All NEET students are chemistry students also. So you will be reading the chemistry as one of the subject. You know that the sulfur atomic weight is 32, that is normally, and phosphorus is 30. But here we have converted this one into radioactive 35 sulfur and radioactive phosphorus 32. Normal atomic weight is here is 32. This is 30. Okay. Now this one plus T2 phage plus E. coli. Here also this one plus T2 phage plus E. coli. When they are cultured in the test tube, they are allowed for centrifugation. After culturing, allowed for centrifugation. So what happens in centrifugation? There will be blending of all the three types of particles. So these both mediums are allowed for centrifugation, so that it will be blending process will be happening here, means mixing. So after blending, then what happens? In one medium where radioactive sulfur is taken, there what happens in the sediment? There will be E. coli plus DNA of phage, T2 phage. Here also the same thing. Here will be E. coli plus DNA of phage, T2 phage. So here we can see on the supernatant, this part which is on the uppermost area is supernatant. The bottom settled one is sediment. Here also this part will be supernatant. This part will be sediment. Here there will be the protein codes which are exhibiting the radiations. Means in this case, in the first one, we see the radiations or the coloring of the particles only in the supernatant. Radiations or colors are seen in supernatant. In case of this, again still the protein codes are present here, but color or radiation when exposed to different when exposed to UV rays, the radiations are seen in sediment. Why? Because sulfur is the element that is present in amino acids. What are sulfur containing amino acids? First priority is cysteine and cysteine, and also methionine. These two types of amino acids have sulfur as one of the element in them. Phosphorus is seen in DNA. We read mostly when we are reading about DNA as diphosphoester bonds created between phosphorus and carbon of 3' and phosphorus and 5th carbon of pentose sugars, etc. as phosphate molecules. Because of presence of more phosphates in DNA, DNA is attaining the acidic nature. So here they have confirmed that in this case where the supernatant is giving the radiations, and here equally, here again we are seeing the radiations in case of sediment. In this experiment, Hershey and Chase has observed the entry of bacteriophage or T2 phage DNA into the E. coli. Here also the same case, but here there is a radioactivity observation. Since DNA is the genetic material, it can be transferred. It has the nature of entering into the host cell for its division. But proteins alone are present in the supernatant. But here, in case of phosphorus 32 radioactive phosphorus containing DNA, DNA is existed or present along with the E. coli, and later this caused the damage of the E. coli cells, and the radioactive T2 radioactive phosphorus which is given for T2 phage now is later seen in the genetic material temporarily of the E. coli, and immediately E. coli has undergone the lysis, and number of DNA T2 phage virulence have been formed in the next stage. So by the entry of radioactive DNA into the E. coli and causing the lysis, and in the next stage or next generation of this, the virulent stages have attained those radioactive natured phosphate molecules. So by this experiment, they concluded that DNA is the genetic material.